Contamination of coffee beans by fungi in Saudi Arabia. Table of contents. Introduction. Poisoned coffee seeds. Materials and methods. Experience and results. Fungal isolation and identification. Results and discussion. Introduction. Poisoned coffee seeds. Arabic coffee is a common beverage in many parts of the world, including Saudi Arabia, which is one of the primary importers of coffee from countries like Brazil, Ethiopia, and Yemen in recent years. Microorganisms can contaminate coffee beans, just like other crops, at various stages of growth, harvesting, processing, transport, storage. One of the most common filamentous fungal genera is Aspergillus. Aflatoxin is a mycotoxin present in a variety of foods such as green coffee beans, ground coffee, and instant coffee. Aflatoxin is most likely produced by Aspergillus segment circumdoti and nigri species in coffee. The goal of this study was to classify the fungal populations and toxins associated with coffee bean seeds collected in Jeddah, Saudi Arabia, and to assess the toxigenic isolates flavus ability to generate aflatoxins G1. Materials and methods. Green coffee beans were collected at random from various regions in the Saudi Arabian markets in 2002. Typically, samples were sent to the lab as soon as possible. Until study, they were collected and preserved at 20 degrees Celsius to prevent the development of toxins or microbial contamination. Fungal isolation and identification. 15 seeds were plated directly on agar plates, and the others were disinfected with 1% sodium hypochlorite for 2 minutes. For the isolation of various forms of fungi, either potato dextrose agar or malt extract agar are used. The developing fungi were counted and classified using macro and microscopic characteristics. The total counts of each fungus species is determined and divided by the weight of the coffee bean seeds in milligrams. Results and discussion. "30 samples of coffee beans were collected from different places and zippers of water content was resolute. The contamination of seeds with different mycotoxins was determined qualitatively. It had been found that mycotoxin production capability by fungi will be limited or impeded by water activity. Increasing water content of the sample increased the quantity of poisons detected. Both 30 coffee bean samples were qualitatively tested for naturally occurring mycotoxin contaminations, and 57% of the samples were mycotoxin-free. Flavus. Aflatoxin production is significantly decreased at temperatures below 25 oC, but is totally inhibited at 37 oC or above. Addition of caffeine to the expansion medium up to 1 gram per liter decreased the fungal growth and aflatoxin G1 production to about 67 and 23%, respectively. Addition of vitamin C to the growth medium up to 4.4 grams per liter enhanced the fungal growth and mycotoxin production by a flavus. Similarly, the result of adding 5 separate plant materials to a flavus growth medium was investigated. Cinnamon, garlic, or both were found to inhibit both a flavus development and afl. G1 performance. Saffron and ginger, on the other hand, had no effect on Aspergillus growth or toxin development, while cardamom inhibited fungal growth while having no effect on the amount of afl. G1 produced. Discovered that adding certain herbs, medicinal plants, or plant extracts to fungal growth media may inhibit or improve fungal growth and toxin generation.